Hey guys, welcome back. This is Lei. Previously, I showed you how to perform alternative splicing analysis. In this video, I'm going to show you how to perform transcript-level alternative splicing analysis. The title is a bit misleading though. There's no transcript-level or gene-level alternative splicing. It's only used to differentiate the tools I used. In our previous tutorial, I used a tool called SPlAdder to identify and quantify alternative splicing events. In this tutorial, we're going to use another tool called SUPPA2 for the events quantification. SPlAdder accepts aligned RNA-seq reads, the BAM files, and the referenced annotation file, a GTF file, as its inputs, while SUPPA2 accepts transcript expression files and a GTF file as its inputs. Remember our previous isoform quantification tutorial? That's where we get the transcript expression files. The outputs of SPlAdder include splicing graphs, splicing events, and event quantification. The outputs of SUPPA includes the event quantifications. SPlAdder can detect novel splicing events, but it requires a lot of computational resources, while SUPPA2 can only identify non-splicing events, but it's much faster and more efficient. So pick the one that fits your research the best. As usual, our first step is to set up the environment. So here we create a new conda environment called SUPPA2_env. After activating the environment, we can download our tool directly from the GitHub page using the git clone command. You don't have to install the tool because all the executables are already in the folder you downloaded. Once the tool is prepared, we can start our first step to generate the IOE files, the index of event files. SUPPA2 is written in Python, so here we call Python, specify the main function, suppa.py. It's in the folder you just downloaded. And call the function generate events to make the IOE files. -i to specify the GTF files you have. Here we use the mouse GTF file. -o is to specify the prefix of your index. Here, that's why all our index file have the IOE prefix. -f to specify the type of your IOE files. Here we use IOE. -e to specify the splicing events you want to look at. Here, we put all the splicing events here to make it easier for your analysis. The interpretation of all these abbreviations are shown below. Skipped exons, alternative splicing sites, something like that. So the output index are named as IOE-A3, A5, AF, all these splicing events. So each event has its own index. This is the inside of the GTF file, the format of it. And this is the format of the IOE file. They're both tab delimited files and can be opened with the Microsoft Excel. But you don't have to know what's inside because you are not going to process them. The tool is going to need them for the following steps. So our second step is to quantify the splicing events. So here we call Python. Here we quantify the A3 events only, for example. So you can do the same thing for all other splicing events. So we call Python, specify suppa.py path, call psi per event function to quantify the events. -i is a IOE file we just made here. We want to look at the A3 events. -e to specify your transcript expression file. So this file is from our previous tutorial, we have two groups over here, class, SPIP, and class. You remember, for each group, there are two samples. Here is the format of our isoform expression matrices. This class, there are two samples. This class, SPIP, there are two samples over here. And the numbers are the TPMs of our isoforms. Notice that the column names are misaligned, but it's fine. The two can solve it by itself. So just leave it like that. -O is the name of the output file. Here we do it twice because we have two groups. So we quantify the PSI, the events, A3 events for each group separately. This is how the two calculate the splicing events. So here, there's a gene that has 3 isoforms. 1, 2, 3. Isoform 2 has an exon skipping event over here. So to calculate this exon skipping event, we use this formula. Expression of isoform 1 plus expression of isoform 3 divided by the expression of all 3 isoforms. So think about it. If the result is 0, that means isoform 1 and isoform 3 are not expressed at all. What does that mean? That means the exon skipping event is 100%. So what if the value is 1? That means isoform 2 is not expressed. That means no exon skipping events detected. So what if the value is between 0 and 1? That means there are partial exon skipping events in our case. That's how the quantification is done. And here is the result, the PSI file is the output. So it shows the quantification of each event for each sample in the class SPIP group. We have another file for the class group. So once we have the quantification for all the samples in both groups, we can perform differential splicing analysis. Here's how we do it. So we call Python, specify the main function, suppa.py, call another function called diff_splice. -m is how you want to calculate the p-value. We use empirical over here to leave it there. -I is a index, the IOE file we just created in the first step. -p, we specify the PSI file we just quantified for two groups, the class SPIP group and the class group. Separate them by space. -e to specify the TPM, the isoform expression matrices. This is for the first group, this is for the second group. Make sure the group's orders are same. -gc means we want to correct the p-values for multiple comparison. -c means if you have more than two groups, it will compare all the combinations among the groups. -o to specify the name of the output. So the output will be a DPSI file. It's basically like our DG table. So the first column is the difference between the PSI between the two groups. The second column is the p-value column. Like here we don't see any significant events. And note that column is misaligned again, so it doesn't matter, you know it. And we have another PSI VEC file generated by the function. It just shows all the PSI values in all 4 samples of the two groups. So once it's done, you can perform the same analysis for all other splicing events. And you can filter them using p-value. And then you can combine them into a single X file, like spreadsheet, and count how many differentially expressed or differential events are there, and then make a pie chart like this one to show you what kind of differential splicing events are there in your dataset between the groups. So this one is made in R, but you don't have to do it in R because it's quite complicated. You can use many online tools. For example, this metachart.com website to make the similar pie chart. You just need to calculate the numbers and put the numbers in each section over here and generate a similar file. And that's all about it. It's not hard and quite straightforward. It just takes some time. And now you can go to my website, go to the corresponding tutorial, and copy the code and try them on your own. And so far we have analyzed, we have performed all the most major analysis for RNA-seq data. So in the next tutorial, I'm going to show you how to upload the analysis and the raw data to NCBI GEO database for publication purpose.